Welcome to this talk about the G protein coupled receptor database, or GPCRdb. My name is David Lorian and I'm the head of GPCRdb. And maybe this is the first time you hear about GPCRdb, or maybe you used it many times. But either way, I hope you will find something new in this presentation and something new in GPCRdb today. So GPCRdb has today about 4,000 users per month and about 4,000 per year. You can find that statistics on the main page, as you're seeing now. And GPCRdb has sections for receptors, but also G proteins, arrestins, biosignaling, drugs and ligands, and structure constructs. And different people come to GPCRdb for different parts of the research. So in GPCRdb, you can find reference data, you can find analysis and visualization tools, experiment design tools, or you can go there and deposit data when you publish. So if we look at the data first in GPCRdb, GPCRdb contains all human non-olfactory receptors, as well as about 28,000 species autologs. The sequence alignments are structure-based, meaning that the positions in the sequence alignment are equivalent in the structural position. GPCRdb also annotates all the structures from the PDB database. They are updated now monthly, and we annotate additional information, not in the PDB database, information that help you select the best template for your study. GPCRdb also build models of the receptors that don't have a structure, and does so in inactive, intermediate, and active states. So there will be a model also if your receptor has a structure on one state, but not the other. GPCRdb also contains genetic variants, so meaning natural mutations that could affect our physiology, cause disease, or the effect of a drug. GPCRdb also aligns G proteins and arrestins. We also have coupling data for those, merging data from different sources. I'll come back to that later. GPCRdb also has a dedicated annotation and browser for structures in complex between GPCRs and G proteins, or GPCRs and arrestins. The biased signaling section is new. We have a comprehensive or near complete annotation of biased ligands from literature, currently amounting to close to 1,300 ligands. They've been tested for bias. We also have pathway effects. So which effect should you target to make a therapeutic? So which pathways should you avoid because they give an adverse effect? We also have a drugs and ligands section, and in there we have a number of drugs and clinical trial agents. We have close to 200,000 ligands. We have ligand site mutations, trying to pinpoint where the ligands bind and how, as well as ligand interactions from structures. And finally, we have a data section for structure constructs, so constructs that have been used to determine crystal structures or cryo-EM structures with GPCRs. We have annotated both the engineering of the protein sequence, as well as which experimental reagents and conditions and methods that have been used. And this slide shows the analysis and visualization tools, or some of them at least, in GPCRdb. For receptors, you can display the topology of residues, either seen from the side in a snakeplot or from above in a helix box plot. Color-coded mutations, for example, that gave an effect in a mutagenetics experiment. You can also investigate receptor relationships using phylogenetic analysis or making a blast against the sequence alignments in GPCRdb. You can also get statistics on how many structures are available for GPCRs and for which receptors. You can also analyze structures, and you can superpose structures also on specific residues, for example, residues in the ligand binding site. And if you have identified a site of functional interest, you can also search that site in the sequence alignment to see other GPCRs that share the same site and therefore maybe can also bind to the same ligand. For G proteins, we have a way to intersect the coupling data to find receptors with a particular profile. We also have interface contacts, so residue-residue interactions across the GPCR and G protein. For arrestins and biosignalings, these are new sections, and we are currently working to develop more tools. For drugs and ligands, we have statistics on drugs and ligands. We also have statistics and detailed information about the interactions between the ligand and the receptor. And for the structure constructs, we actually can analyze the modifications in great detail, both the truncations, the fusions, deletions, and stabilizing mutations that have been done. The third slide here on the experiment design, there I want to highlight three main tools. The first one is new and called sequence signature tool, and you will find it in the receptor section. This is a tool to identify functional determinants. So if you have receptors that share a function and other receptors that lack the same function, this tool can help you to identify the residues in the sequence that actually determine whether other receptors have this function or not. For ligands, we have more than 4,000 mutations from literature. Also, as I mentioned, we have interactions from ligand receptor complexes in structures. So using these data, we have compiled a tool to quickly design new mutations. And these data are inferred to other receptors that don't have data yet, meaning you can target any human GPCRs with mutations to find the ligand binding site. And the construct design tool is to make new structure determination experiments. If you do crystallography, probably you're interested in all the type of modifications. If you're doing cryo-EM, maybe you'd like to know at least about N-terminal truncations, where to put maybe a single peptide to increase the expression, and find some experimental conditions and reagents. And this is the last slide before I go online, which shows you something about data deposition in GPCRdb. And actually, I'd like to start on the right side, because for ligand binding sites, we have more than 34,000 mutations. And these have been annotated from literature from other groups. For example, when they've written a review about a particular receptor family and its ligand interactions. But something we'd like to see more of is when authors deposit data when they publish their manuscript in a journal. So if you have mutagenesis data and you're going to publish it in a journal, it can be a good idea also to publish it in GPCRdb. If doing so, you can compare your data to already published data. You can visualize the data in our diagrams and use them for the publication. Also, maybe more readers will find your data. And for the G protein coupling profiles, the golden reference for a long time has been the Guide to Pharmacology database, which has couplings for 253 GPCRs, and these are curated by pharmacology experts. However, these data are only for the G protein families, whereas in the last 2 years, there's come two systematic data sets with high throughput assays investigating individual subtypes of G proteins for a large number of receptors. And GPCRdb is integrating this data in one browser, so you can easily find receptors and their G protein coupling. And that reminds me, now it's time to go online to show the last two sections. The Citus page is where you can read up more about what to find in GPCRdb and how the data was produced. You will see a highlighting of the main reference, but if you select any page in GPCRdb, you will also find a highlighting of the specific reference for the page you were using. And finally, GPCRdb wants to be a community resource. So in the Join Us section, you will find information about how to help with data annotation, maybe publish together, how to join us and support the data development, which has open source code, or how to make scientific collaborations, either depositing large data sets in GPCRdb or collaborating using our analysis tool on your scientific data and studies. And finally, ways to share and find information about the database, as well as ways to give feedback. So if you're using GPCRdb and you're missing a functionality, then please contact us at any time. I hope that this introduction, which was only a brief introduction, has given you a few new ideas. And please go and try out the database, find the sections of most interest to you, and let us know what you think.